So I'm Bella. I'm an undergrad here at the University of St. Andrews, and today I'll be talking to you about some work I did in the labs of Martin and Stefan's labs about how movement coordination is achieved by Tribolium castaneum larvae. And, well, we've already received a little spoiler, but before that you might have been wondering what the heck is that and why should I even care? Well, for the first question, Tribolium are a beetle species, and as you may have guessed from their name, they eat and live in flower. And importantly, we'll be focusing on the larval form here, which have segmented bodies and a segmented equivalent to the spinal cord, but also 3 pairs of thoracic legs. I also want you to take note of these little protruding structures in the most posterior segment called pygopods, because they will be important later. As for why you should care, Tribolium are a major agricultural pest, and although they are currently mainly found in the global south, global warming is already promoting their spread to the northern hemisphere. And countries that are producing crops susceptible to infestation are currently and will continue to suffer great economic losses. So there is a major precedent to find ways to control their spread. But also from an evolutionary perspective, Tribolium are also a really popular model in studying insect development, since it has a more ancestral lineage and is perhaps more representative of other insects than, say, Drosophila. And after its genome was fully sequenced in 2008, this created a great incentive for comparative studies to see how genes vary between insects and what this might mean for the functions of their vertebrate orthologs. And indeed, a lot of comparative work has been done, especially by some labs in Germany. For example, they began to characterize the Tribolium neuromuscular system and found a lot of similarities between the muscles of Tribolium and Drosophila larvae. But in spite of all this genetic and developmental work, Tribolium are still heavily understudied in neuroscience, and especially in our field of motor control. And coming from 2 labs that work a lot with Drosophila, we became quite interested in this thing that we affectionately started to call a maggot with legs. And taking everything I've just said in mind, there were a lot of interesting questions that we could begin asking. For example, how is larval locomotion actually achieved? And how does the larval CNS coordinate thoracic and abdominal segments for adaptive locomotion over unpredictable terrain? So those are the things that I'll be covering today. As to how they achieve locomotion, we found that larvae use a bilaterally symmetrical gait to walk. So the 2 legs within a segment swing at the same time, and this starts from T3, which are the most posterior thoracic segments, and it propagates up to T1. Now this is quite different from most hexapods like adult Drosophila, which more commonly use a alternating tripod gait. And insects that do have this bilateral symmetric gait are often those that traverse unpredictable terrain, like the desert dung beetle, for example. So it's very possible that this kind of wave gait is the most energetically favorable for terrains like sand and flour that are constantly shifting around. We also find that limb coordination increases with speed. So in the fast animals, as I've shown before, you get this bilateral symmetry, but in a slower animal, you often get this incoherence and asymmetry within leg pairs. But you can still just about see this propagation from T3 to T1. And indeed, when we plot the phase difference between segments, you see the sequence of T3 to T1 is preserved regardless of speed or frequency of locomotion. But the phase differences between legs within a segment become greater in slower animals. That is, they are less likely to swing at the same time. And lastly, when we analyze the swing and stance relationships across speeds, we see this decrease in stance duration with increasing speed, which is the typical relationship you see in many other models like flies, mice, rats, cats, just to name a few. And after characterizing gait kinematics, we were also interested in understanding the extent to which the thorax and the abdomen contributed to locomotion. So to do this, we surgically severed the connectives between A1 and A2, the ganglia, which effectively disconnected the abdomen from the brain and thoracic segments. And since these animals are also capable of climbing, we tested them on both a flat arena and on a challenging overhang to see how removing descending inputs to the abdomen would affect both normal and challenge locomotion. We then saw that compared to controls and shams, Severed animals are heavily impaired in locomotion and generally much slower. And when we analyze the gait kinematics under these conditions, we surprisingly find similar patterns as normal larval walking. So severed animals can still walk following this T3 to T1 sequence, and also follow the same trends in swing and stance phase relationships as controls and shams, but are just generally slower. So from this, we thought that maybe the inputs from abdominal segments may not be required to coordinate thoracic leg movements. However, we also noticed that severed animals also perform far worse on the overhang, so on climbing, and controls and shams could all climb with no problem, but most severed animals either made no progress or fell off almost immediately, like this little guy. So we thought that maybe descending inputs to the abdomen may be required for more challenging locomotion. Now, if you look closely at the sham animal climbing, you'll notice these little pylopod structures I was talking about before. Now, these are exclusively recruited by controls and shams during climbing, and they involve extending and planting these pylopods into the substrate. And when we plot a histogram to show when legs are swinging in relation to these pylopod plants, we see that swing onsets align with when plants occur, which is at 0. So we believe these to be anchoring devices that stabilize the animal while the legs are lifted off the ground, rather than a mechanism for, say, increasing speed. And we think this because planting the pylopods actually comes at the cost of losing speed, which is, we think, sacrificed to increase stability and lessen the risk of fall. So although disconnecting the abdomen does not disrupt thoracic coordination in normal walking, it seems to play a greater role in challenge locomotion, especially by enabling the recruitment of these pylopods. And finally, to see how these effects translate to navigating the current natural environment, we also look to see how these manipulations affected tunneling ability. And normally, larvae tunnel really fast, and in the time that we just spent talking, controls and shams are almost completely submerged. But quite strikingly, you'll notice that even after 2 minutes, if you give it time, severed larvae just could not tunnel, and not for lack of trying, because you see the legs and head constantly moving, but they're just not making any progress. So the abdominal segments appear to also play a crucial role in enabling tunneling, which for this animal is the main strategy for feeding, development, reproduction, and infestation. So our findings may also have important implications for pest control. So to summarize, we have characterized the gait of the Tribolium larva and found prominent roles for their abdominal segments, especially those pylopods in challenging locomotion. And we think the abdomen could potentially be communicating sensory information to the brain, for example, regarding how much the abdomen is in contact with the substrate, which the brain may then compute and send descending signals down to plant those pylopods for stability or to generate those pushing forces that are required to submerge in the flower. And in the future, to properly understand descending and ascending inputs between the brain, thorax, and the abdomen, we're looking to characterize the cell types within these segmented ganglia, and especially those that project posteriorly to those pylopods. With this, we could then perform cell type specific manipulations with the great genetic tools that are available in Tribolium, like RNAi and also Cal4US, and plus, not to mention all the potential electrophysiological work that we could do, for example, recording the rhythmic outputs from these ganglia. So in short, there's plenty of work still to be done. And everything I've covered today and much more was published recently in the Journal of Experimental Biology, which you are more than welcome to check out and so chat to me about. I'm pretty easy to find. Just look for some short person, also always dressed in black. It's usually me. A big thank you to the IVANS undergraduate research team for funding this, and to my lovely supervisors, Martin and Stefan. And of course, thank you all for listening. I'm more than happy to take questions.
Being able to